if anyone has anything. All right. Well, if not, I can go ahead and introduce our speaker. So I'm very excited to introduce Dr. Lindsay Constantini. Dr. Constantini got her bachelor's and master's degrees from the University of Connecticut in molecular and cell biology. And she went on to do a PhD at Albert Einstein with Dr. Eric Snapp. And I think it was during her PhD that she started some collaborations with some virology labs and discovered an interest in virology. And so that led her to want to continue into a postdoc. And so she came to UNC Chapel Hill, which is where I got to meet her, and we overlapped for just a little bit in the same postdoctoral Spire program. And while she was there, she worked with two very excellent virologists, Dr. Blossom Damania and Jack Griffith. And then she has since taken her research on to North Carolina Central University, where she looks at molecular interactions involved in human herpesvirus DNA replication. And so I think with her, she also brought a lot of cool microscopy and some electron microscopy stuff to Central, and has some really beautiful images that I'm excited to take a look at. And so with that, we're excited for your talk, Lindsay.
Well, thank you. And I'd like to also thank the extension of the invitation to speak with you all today. I'm excited to share some of the work that I've been able to continue from UNC, and also I started at NCCU about 3 years ago, and so we've been continuing that work. So as we've already heard, I've been integrating my past experiences. So my background is primarily cell and molecular biology, but I did make the switch into virology during my postdoctoral fellowship. And I continue to bridge those two worlds and the intersection of where cell biology meets virology. And one of the common denominators for me has always been the inclusion of microscopy. I've always loved microscopy, whether it's live cell microscopy or the newest types of microscopy that I've been including, which I'll be talking about today, electron microscopy. So my lab is kind of the intersection where we kind of combine all of these different areas to really study the cellular and molecular mechanisms of oncogenic virus DNA replication. So for the purpose of my talk today, I've kind of broken it up into 4 areas. So the first is just going to be a quick discussion about using electron microscopy to really visualize protein-protein and protein-DNA interactions. Then I'll give you a little bit more background into the virus that my lab studies as a model virus, which is Kaposi sarcoma herpesvirus, abbreviated KSHV. I'll talk more about the KSHV DNA replication machinery, and then how I've been using transmission electron microscopy methods to visualize the purified KSHV core DNA replication proteins and the proteins in DNA that are important for governing the initiation of viral DNA replication. So I'm sure many of you have probably seen a slide like this before. One of the things that draws me to microscopy is that ability to see things. So we all can obviously look around and see things with our eyes, but if we want to look at those really small things, we need the aid of a microscope. And depending on what you're trying to look at, depending on if you're in the realm of nanometers or microns, you might need to use an electron microscope. And so in this part of my research, I focus a lot on the smallest things on this scale, down here in the protein range and the DNA range. So in order to conduct the studies and ask the questions that I'm currently asking, I need to use an electron microscope. And I'm just showing you here just a wide selection of some of the types of EMs and what are the things that we can visualize using specifically transmission electron microscopy. So over here are just some representative TEM micrographs. So starting up here, we're at the micron level, and then we can go all the way down, oops, sorry about that, to the nanometer level down here. So at the cellular level, at the micron level, we can look at individual cells. And electron microscopy historically has been used a lot to understand the ultrastructure of cells, organelles, how cells are organized. We can go a little bit further. So in panel B, this is actually the nucleus of a B cell that's infected with KSHV. And those little punctate structures that you're seeing are actually the start of the viral DNA replication centers, where all of the lytic DNA replication is starting to go on. We can also look at subcellular structures like the cytoskeleton, individual organelles or viruses. And then what my focus on today's talk will be is at this highest resolution, at the nanometer level, where we're looking at individual proteins and individual DNAs. And I can look at things that are typically, in terms of DNA, a piece of DNA that's at least 100 base pairs or longer. DNAs that are proteins that are at least 10 kilodaltons or bigger. And this is enabled because I can use an electron microscope that can give me the magnification of 50 to over 750, 100,000 times magnification. So before going into the data, I also just want to talk a little bit about the power of what microscopy can give us in terms of this idea of thinking about different assays and different methods in two realms, this idea of an individual versus a population method. So if we have this hypothetical population of cells, I've drawn them here in like this cartoon diagram, and as you can see, they're different colors. So we could do a traditional method where we do a bulk analysis where we might average them all together and then do a certain readout. So in this hypothetical experiment, let's just say we're doing mRNA transcript levels. So the y-axis will be some sort of level of expression. So if we take this bulk population of cells, put them all together, we're going to get an average readout for whatever transcript we're interested in. However, there are some methods, microscopy being one of them, that we can look at individual cells and we can sort them. So what if we then broke up this population of cells into individual groups, in which case we have all the purple cells in one group, all the green cells, all the blue cells, and all the pink cells. And then we looked at the individual transcript levels for each subpopulation. And what I hope this cartoon diagram kind of gives you the feel for is if you look at the cells individually, or the population of each color or each category of cell, you can see that there's variation. Like all the cells themselves are not having an identical transcript level, but if you use more of a population or an ensemble method, you only would get the average. And sometimes the average isn't always indicative of an actual true population. In this particular example, you can kind of see that the average is pretty similar to what these purple cells are showing, but in instances where we've subdivided into the blue cells, clearly their expression is much lower than the average. And vice versa, with the green cells, their expression would be higher than the average. So all this to say is I believe that science obviously requires multiple approaches to be used in concert to get the most powerful answers. And together you can get a robust and comprehensive picture. And so in the cell biology and microbiology realm, you might think of some typical population methods as listed here. So things like gels or blots. So you can do things like EMSAs, which I'll talk about a little bit later, or Western blots, in which case you're looking at a band that represents the most abundant species. In addition, like I gave you that example with the hypothetical different colored cells, you're looking for a relative signal above a certain threshold, and sometimes it's the average. And then also there's other plate reading and other types of ensemble methods that will allow you to look at an overall change above a certain threshold. And although these are really robust methods that are typically used in many different research labs, there are certain limits. And like I used in the example I just showed you, sometimes the average might not be completely indicative of what's happening in each single cell or each individual molecule. So in that same realm, some common methods that you might have been familiar with that allow us to look at single cells or single molecules include microscopy. So for instance, whether it's live cell fluorescence or EM, which will be what I'm talking about today, you'll be able to see variation, whether it's cell-to-cell variation or molecule variation. In addition, you could use a flow or a fact-based method, and that allows you to look at individual populations that you discern based on the characteristics that your experiment calls for. And although these have their strengths, there are limits to this method as well. And I think that just goes to say that in science, no matter what method you're trying to use, there's always going to be advantages and disadvantages. In this case, when you look at single cells, you're a little bit limited in terms of the processing time and the computational power you need to have in order to look at and pull out those interesting differences. So for the purpose of this talk, we're going to narrow our scope a little bit more into EM. So I particularly use transmission electron microscopy. I use it to visualize protein DNA interactions, looking at where proteins bind to specific DNAs, as well as DNA structures and complexes that form from pure, in the case when I'm looking at purified proteins. So here's an example of how you can look at different populations using different methods. So on the left-hand side is a representative electron micrograph. For the purpose of this slide, all you need to know is that this is just a viral protein. It happens to be a processivity factor. I'll talk more in detail about the proteins and their functions a little bit later. But what you can see, I hope, is that from this electron micrograph, we're just looking at purified protein alone. And so these arrowheads are going to show you these different round, like Cheerio structures. There's also this structure down here that kind of looks like it has 4 petals. And then the arrowhead over here is actually denoting just one small protein over here. So just by looking at an electron micrograph of purified proteins, we can visualize that there's actually different conformations. If we then try to examine the same protein using a gel-based assay, we might not see all of the differences that we're able to observe if you use a microscopy approach. So just briefly, over here we have a semi-native gel. Here we have a ladder just to denote the approximate molecular weights of these bands. In which case we have BSA as our control. So BSA is a protein that is known to oligomerize, so it can form dimers, trimers, etc. It happens to be approximately the same size as the protein of interest that I showed you in this electron micrograph. And so we could use it as a protein standard comparison to be like, okay, well, what does our protein do when you put it into the same type of gel? It also confirms for us that we're able to maintain certain interactions that we're looking to visualize in the gel, the protein-protein interactions. So in this line right here, it's just the same protein that's visualized in that electron micrograph. And what you can see is a predominant band up here. And if we're using BSA as a control, we think that that's usually either a hexamer or a pentamer. So it's a high molecular weight complex, meaning 5 or 6 of these proteins are actually coming together to form this complex. If we look at the same gel with a little bit higher exposure, we can start to see another band kind of pop up down here, which based on the sizes of the ladder we're using as well as the BSA comparative standard, it could be the size of a monomer. So yes, with a gel-based method, we can certainly see that there is protein, and this protein has the propensity to interact and form these larger complexes that migrate higher in the gel. However, what we're missing is all of this different information about whether or not it has 4 maybe monomers together, or maybe this could be a hexamer where there might be 6 of those monomers together. And so all of this to say that we can use EM for not only qualitative, so we can look at an image and what are we observing? We're observing multiple structures, but we can take it a step further and then count the frequency of the species observed. Is there a predominant population? So in this case, this is obviously just one representative micrograph. If we were actually doing this, and like I'll show you in some subsequent slides, we would look at hundreds of molecules. But let's just say this is just representative of what we would see in those 100 molecules. You would say that this Cheerio or donut-like structure is clearly the most predominant species that we're observing, whereas these other species, the monomeric or maybe this tetrameric structure, might be in the minority. However, we're able to visualize them so we know they're there, and then that can lead us to design other experiments that might help us understand its function. So what if we then incubated this protein with DNA? Would one of these species become the more predominant species? In addition, I'll show you some data later where we actually can quantify the dimensions and understand a little bit more of the function based on the size and dimensions that these proteins are forming. So the next thing I want to talk about, which will lead into the KSHB data that I'm excited to share, is this idea of mapping DNA positions. So we all know that you could also ascertain some of this information using, for instance, a IP where you pull down the DNA and protein together, then you can do some digestions and you can send the DNA out for sequencing and receive the predominant sequence of whatever the location of where that protein is binding. But what we can do with EM is we can literally visualize protein bound to DNA. So in this micrograph, this spaghetti-like structure that I'm kind of highlighting and outlining right now is literally a piece of DNA that we're interested in looking at. We know the sequence of it and we know the size of it. And then we can also co-incubate it with different proteins. And just in this picture, you can see 2 different proteins bound to this piece of DNA. There's a larger protein bound more internally, and then there's a protein that's bound to the end. So we can see, is there more than one protein, the size, the relative size of the different proteins that we're looking at, all in relation to how it interacts with DNA. A traditional assay that allows you to ascertain whether or not protein and DNA are interacting is an EMSA. And so this is just a representative EMSA for the purpose of this slide. All I want to go through is just the basics. And so in the first line right now here, we have a piece of DNA alone. So we've just incubated DNA and ran it in gel, and we see a band at a predicted molecular weight. If we then do an experiment where we co-incubate the protein in the DNA, we see a shift. And that shift means that the protein and DNA are likely interacting. That complex is going to be larger than the DNA alone. Therefore, we're going to see that shift in this higher band in the gel. We can also repeat this for 2 other pieces of DNA. They happen to be smaller pieces of DNA because they run further in the gel. So lane 3 and 5 are just the DNA alone. When we incubate the DNA, each DNA with a protein, we can see this shift. We see the appearance of this higher molecular weight band, which again indicates that protein and DNA are interacting because it's a larger complex, therefore it's going to run slower in the gel. But what this gel can't tell us is how many proteins are bound and where are these proteins bound. And so we can do, again, qualitative analysis on our EM micrographs, where we just look at the number of proteins per DNA. That's pretty interesting in itself. We can also look at the relative size of the proteins that are bound, and we can go a step further and do some quantification where we're going to look at the actual position or location, as well as the area of the protein that's bound to it. So that's a little bit of the background of where the methods are going to go. But before we dive into the actual data that talks about the KSHB proteins that I'm interested in, I just want to give a little bit of background about the virus that is the model virus in my lab. So Kaposi-sarcoma herpesvirus might not be a virus that you've heard of. It's also called Herpesvirus 8, HHV8. It's a gamma herpesvirus. In humans, it's a virus, a herpesvirus that infects humans. If infected, it will infect your endothelial, epithelial, and B cells. It's one of the cancers, one of the viruses that is associated with the onset of human cancers, especially in immunocompromised individuals. It can cause Kaposi-sarcoma, which is what the virus is named after. And it's also associated with a few B-cell lymphomas. And like all herpes viruses, there's a lifelong infection. And so this electron micrograph is just an example of new virions being assembled in the nucleus of a KSHB infected B cell. So again, we can look at the ultrastructure and we can start to see some localization information about the virus and cells we study in the lab. So for those who aren't as familiar with oncogenic viruses, it's predicted that about 15 to 20% of human cancers are caused by viruses. The virus that I study is an example of a herpesvirus family member that is that can cause cancer. There's about 8 known viruses that have been identified. Epstein-Barr is another one. You probably are more familiar with human papillomavirus, or HPV, and how that's been highly associated with cervical cancer. Also, both hepatitis B and C are highly associated and can be the causative effect of liver cancers. So they're just subdivided for the purpose of this slide into the 2 major groups, whether they're DNA or RNA viruses. There are viruses in both family subtypes that have been known to induce human cancers. And just a little bit more about the human herpes viruses. So those herpes viruses that can infect humans, they're also subdivided into 3 families. So the alpha subfamily, which is up here, are probably the herpes viruses that you're most familiar with. So herpes simplex 1 and 2, those are the herpes viruses that are known to cause the lesions, either cold sores or genital lesions, as well as varicella zoster virus, which is the virus that is known to cause chickenpox and shingles. In the gamma herpes virus family, we have 2 viruses. We have Kaposi-sarcoma herpesvirus, which is the virus of topic today, as well as Epstein-Barr virus. And you might have been more familiar with Epstein-Barr virus. It's very related to KSHB. It's a virus that's known to cause mononucleosis, or mono, and about 95% of the population is predicted to be infected with Epstein-Barr virus. And if you remember from the last slide, it is one of those viruses that can be associated with the progression of human cancers, but normal individuals who have an intact immune system and are not immunocompromised, the likelihood of developing cancers associated with these 2 herpes viruses is very low. However, all herpes viruses have the common trait of having a lifelong infection, meaning once you're infected forever. And the way that herpes viruses actually do this is they have a biphasic life cycle, which means they just have 2 phases. So after primary infection, so once you're infected with the virus goes into what's called latency. And that's the dormant phase. So it kind of goes under the radar and it tries to be covert and not be detected by the virus. However, during the lifespan of the infected host, there are certain events that will cause the virus to reactivate. The exact causes of that are still being determined. My lab also works on a project at the cellular level where we actually examine some environmental factors that actually can lead to the reactivation of KSHB. But it's not well worked out, but it does happen sporadically through the infected individual's life. And this causes the lytic, or active phase. During the active phase of viral replication, you're going to get new viruses produced. And in order to produce those new viruses, viral proteins need to be made, the viral genome needs to be copied. These components need to come together to make the new viruses that then can be released from the cells and go on to infect other cells in the body or go on and infect other individuals. So our lab primarily focuses on the DNA replication step. So without any of these steps, if any of these 4 steps are unable to be completed, you don't get any new viruses. So we specifically look at the components that are already identified that are known to be involved in viral DNA replication. So you can break it up into two categories. The first, what DNA regions or DNA sequences are important for the initiation of DNA replication. And like all DNA replication, KSHV's genome encodes two origins of replication, lytic origins of replication. They're just denoted left and right. I have a schematic on an upcoming slide that we'll talk a little bit more about that in a second. In addition to the relevant pieces of DNA where the DNA replication will initiate, you're going to have proteins come in to actually do the jobs of replicating that DNA. And so in KSHV, there is known to be 7 essential DNA replication proteins. KSHV encodes its own machinery. So during the lytic phase, it just upregulates and expresses all those proteins, and the genome is present in these infected cells. And so it can just go on and start making mass copies of its genome to then go on to make new viruses. So like I said, the KSHV genome encodes 2 lytic origins of replication. In this diagram down here is just an example of like a cartoon representation of the entire genome. So what you can see from this hopefully is on average, KSHV is about 170 kilobases long. It happens to be a pretty large DNA virus compared to human papillomaviruses, which are typically small DNA viruses. And that's why herpesviruses encode all of their own machinery, and they encode hundreds of different genes. So in general, it's considered a large double-stranded DNA genome. It can have 2 major conformations. In the virions and during the lytic replication, there are the linear conformation, and when the virus is in its dormant phase, the genome becomes circularized. And just here is just an electron micrograph where I was able to take different KSHV virions, which are these bright white spots, basically burst them open, and then they extrude out their DNA, so you can really get an appreciation for how large the genome is and how much has to be compacted inside those virus particles. So my lab, like I've said before, is focused on an in vitro system, meaning that we want to look at the purified proteins and the purified DNA. So we have a system set up where we can express and purify the proteins that we're interested in studying. In addition, the sequence of KSHV is known, so we can subclone or synthesize the sequences that we're interested in examining, amplify them and isolate them, and use them in concert together to look at the interactions between the DNA replication proteins and the important DNA sequences. So ultimately, the long-term goal that we're hoping to pursue is this idea of creating an in vitro system to study DNA replication. So can we do this in a test tube? Can we put all of the essential parts and be able to synthesize more viral genomes? If we can, that provides a great tool to then go back and look at how can we disrupt it? What are these structures that form the DNA that gives us indications about how the DNA replication proceeds? There's a few theories on that. So if we had this system set up, we would really be able to ascertain a lot of those questions about herpesvirus replication as well as potentially other viral replication mechanisms. So ultimately, before we can do all of that, we first need to understand how do the KSHV DNA replication proteins organize at the viral origin DNA. So here's just a cartoon diagram. This is the cartoon that I showed you before. The entire genome in these light blue areas are the 2 locations of the 2 different origins, the left and the right. And I'm just showing you kind of a blowed up version of just that, or the right origin. And I'm showing you hypothetically 5 of these 7 or 8 essential DNA replication proteins. And what we hope to do is figure out the order of proteins and which proteins are essential to go from this closed conformation at the origin to this open conformation of the origin that would permit DNA replication to proceed. So we want to look for where on the DNA do the proteins bind? How do the proteins interact with each other or with the DNA? And then what order must the proteins be recruited to form this pre-initiation complex? Because all of this information together will allow us to potentially identify novel replication inhibitors. Because remember back in the slide where I talked about the life cycle, our goal would be to stop lytic replication. DNA replication is one of those key steps. So if we can stop more genomes from being made, we're going to be able to stop KSHV from spreading. So our lab has started work on characterizing the individual proteins. And today I'll be able to share the story that we have for 2 of the proteins, OR50, which is the origin binding protein, and OR59, which is the processivity factor. So I've already given you a couple examples, but just to give a little bit more detail into the methods that we use. So I've already proven to you that we can isolate and purify and visualize different DNA sequences that we're interested in looking at. So again, just for reference, this spaghetti-like structure is in fact the viral DNA sequence that we're interested in looking at. And as you can see from this slide, they might not fall on the EM grid always in these, oops, sorry about that, in these lines. Sometimes they're going to fall on, they can form this loop. So I haven't really talked about much lately, but we could see if a protein causes the DNA to form a loop, we can see the geometry of the protein, of the DNA change in the addition of these other proteins. In addition to just looking at the overall architecture of the DNA, we can also preferentially label one end of the DNA. So here we're using biotin and streptavidin labeling. Again, we know the sequences. We can design a situation where we know we can label one end of it, and that will give us orientation. Because clearly, this DNA right here looks identical at this end and this end. We have no idea if it's the 5' or the 3' end. We don't know what's going on. So in addition to the DNA, we are interested in studying the proteins of interest. And this is just a little bit of background about OR50. It's also known as RTA. It's based, for the purpose of this talk, it's the origin, it's a viral origin binding protein. It's essential for viral reactivation. It is known to recruit the DNA replication proteins. It's important for not only initiating lytic DNA replication, but it also has a role in initiating transcription. It also has known binding partners, so it already is known to bind the DNA, and it also has another protein binding partner, which is the protein we're going to talk about in the second half. So just shown here, we've now mixed together the protein and the DNA, and so where these arrows are, you can clearly see that the protein is interacting with the DNA. There's also some proteins that are not bound to DNA, and you can clearly see that there are some DNA that haven't have any proteins bound to it. So there's a little bit of working out the correct stoichiometry in order to make sure that you can visualize these interactions. But at this point, the published literature says that OR50 should bind to the DNA, the proteins bind to the DNA, and we're able to visualize them. So now we can go a step further. So we've been able to recapitulate what we predict should happen with the proteins, the purified proteins in DNA. So now the first thing you want to do is, look, can we get positional information? So in that one of those first slides, I showed you that we have two different proteins, and we actually controlled streptavidin binding to the end. So what we did is we know the sequence of this DNA. We incorporated a restriction site that would allow for a C overhang. We can come back in with a polymerase, and we can use a nucleotide that has a biotin label. And the biotin's only going to label one end of the DNA that we're interested in. So then what we can do is we can use the end that's labeled as a marker to then compare the distance between the protein and the DNA, the streptavidin and the protein of interest. So for instance, we can measure the entire length of the DNA. We can also go on to measure the length of from the streptavidin label to the beginning of the protein of interest that we're interested in studying, the unlabeled end of the protein, and we can do all sorts of different measurements that in the end, because again, we know the DNA that we're putting in and we know the sequence and size, we can convert the nanometer length measurements that we manually take by tracing to the base pair position in the lytic origin sequence. And so it just so happens that the protein the OR50, the origin binding protein shown in this picture, if you map it back and you convert our nanometer measurements to base pairs, it actually binds to this region right here. So what we can do is we can repeat this hundreds and hundreds of times. It is, like I said in the beginning, sometimes these microscopy-based methods tend to be a little bit labor intensive, but some of the data that we get is really exciting and surprising. So just shown here is on the y-axis is frequency. So the higher the bar means that there were each DNA protein interaction that we mapped, there was more proteins bound at this site. Whereas when you're down here, we only found one protein out of the hundreds of proteins that we measured that was at this location. So if we do that and we can map the frequencies, and the higher the bar, the more frequent that there was a protein bound at that location, we can start to identify preferential binding sites of this protein on this particular DNA. And so in this cartoon diagram on the bottom, I have three regions. I have A, B, and C. So, and then I have green and purple. So there are predicted OR50 binding sites that have been published using more traditional methods, and those are highlighted in purple. So published studies show that OR50 should bind to this location. If you take that consensus sequence and make it a little less stringent, you can identify a very similar sequence right here. It's not 100% identical to the previously published, but it's similar enough that we're like, okay, this looks like it could be a similar sequence of nucleotides. Maybe this could also be a binding site. And so the color bars here just show you where the higher frequencies are. So from here to here corresponds to about from here to here, and so on. So what we can do is we can take what's already known, and then we can map our frequency data. And the most exciting thing that we've been able to show so far is that the site that has the highest abundance is not a predicted binding site. It's a novel binding site. So using the method that we're using, we're able to not only see the predicted, and so we do see a higher frequency, but when you compare to region C, which has the highest frequency, we're now really able to identify that, yes, it can bind at higher frequencies to multiple places, but look, the most abundant site is not already known, and it's a novel site, which is very exciting. In addition, just as proof of principle to make sure that our method does in fact measure what we think it is, we've also been able to use CFOS, CJUN, which are pretty common transcription factors. They're also very well studied. The AP1 site is very consistently bound by these proteins. And in the origin of replication, there's actually 3 AP1 sites. And again, I've denoted the exact location of these AP1 sites with these purple bars. And if you look at the highest frequency binding sites, we can clearly see using our method that we do see this high frequency at this location, and this location, which again confirmed for us that not only is our method validated, but for well-studied CFOS, CJUN transcription factor that has a well-known site, we can clearly see binding and measure binding at all 3 of the sites. The next part, we kind of take it one step further. So now that we say, okay, we can use this to map location and we can identify novel binding sites, can we say something about the size of the protein that binds to the DNA? And so up here again is just a representative electron micrograph where we have, here's just an origin of DNA with no protein bound. Up here we have another origin DNA sequence, and you can see right in the square, there's a protein bound to it. So what we can do, just like we measured the length in nanometers of the length of the DNA, we can measure the perimeter of the protein and then extract the area for that molecule. So shown here in this blue little trace, we can use software, we can trace it, we can then equate that to an area of that molecule, and then we can make a dot plot. So all of these little blue dots are each individual OR50 molecules that we have painstakingly traced and measured in order to get some more interesting data from it. And as you can see, there's variation. So there's not just one size that we're visualizing it at, and we do see some variation, but you can clearly see that we have a mean and a spread that's indicative of a certain size. The cool trick that we've been able to use is this idea of using protein standards. So much like that BSA example I used when I showed you that semi-native gel, there are proteins that are known to be very regular in size. They're commonly used to calibrate different equipment. We can use those same proteins, look at them using EM, and come up with a comparison or a standard. And so what we can do now is OR50, the origin replication protein that I was just talking about with the position, has a predicted molecular weight as a monomer at 75 kilodaltons. So conalbumin, which is one of those molecular proteins that are used for calibrating different equipment, it's very regular size. We can do the same thing. We can measure the perimeter and get the area. And so all these little red, pink, or purple, however fuchsia colored dots would then correspond to a data point on this graph. So then we can get the spread of the area of all the conalbumin molecules visualized and then manually quantified using EM. We can do the same thing for another molecule, alcohol dehydrogenase. It has a predicted molecular weight of 150. And again, I'm indicating that we traced and made the area of all these little green circles. Again, we plot this on a dot plot, and you can see the spread of all the areas of all the molecules that we represent, that we measured. So we have a molecule that would be indicative of the predicted monomer of OR50, and then we also have a molecule that would be close to the predicted size of a dimer of OR50, alcohol dehydrogenase. So we do this for all different types of conditions. And so this graph right here actually splits up into 2 major conditions. So what is the oligomeric state of OR50, which is that KSHB origin binding domain protein under different circumstances. On the left side of the graph, oops, let's go back. The left side of the graph right here, this is all proteins that are by themselves. So isolated proteins not in the presence of DNA. And again, we plot each dot, and you have a dot plot so you can get a real easy idea of what the spread is. You can see the average. Again, it's very similar to the data that was on the other graph that I just showed you on the other page. We have alcohol dehydrogenase, which would be equivalent to a dimeric form of OR50. Conalbumin would be the monomer. We use streptavidin as just an internal control We know that it's smaller than conalbumin, and it's just another internal control because that's the molecule that we use to label the ends of our DNA to give directionality. On this side of the graph, what we have is what if we incubate together the protein and DNA? So these are the sizes of the proteins that when they are bound to DNA. And so to make it a little bit more of a confusing graph, I'm adding some more lines. And so up here where you have the large dashes, this just represents the mean plus the standard deviation of what we would consider our dimeric protein, the alcohol dehydrogenase. So basically we went over here, we have the mean here and the standard deviation. So we just set those as our parameters. We did the same thing with conalbumin, the smaller dotted line. Again, we're using the standard deviation of the mean to set the parameters of what a monomer and around 75 kilodaltons would be. We can then use that and figure out what percentage or what population of our protein bound to the DNA falls into each one of these categories. And if we plot that as just a simple pie chart, we can subdivide our protein into these two categories, the protein alone, which was on that left side of the graph, or when we have protein bound to DNA. And what I think is pretty cool and interesting is that when you're just looking at the purified protein alone, you have about a 50-50 split. It can sometimes be a monomer and it can sometimes be a dimer. Okay, so we observed both. However, in the presence and being bound to DNA, you see about twice as much of the protein is formed in this dimer structure. So there is still some monomer. However, we have a large portion of it that is forming this dimer, which is now we're going a step further. Not only do we know OR50 binds, we've been able to identify a novel binding site, and we're seeing that it has a preference to be a dimer when bound to DNA. We take our analysis one more step, and this is probably our most exciting data. So do monomers and dimers bind to different places on the DNA? So again, we're doing kind of a multivariable analysis. So we can take the information that we got from the dot plots that basically tell us monomer and dimer based on the two standards that we're using, alcohol dehydrogenase and conalbumin. We can take the frequency plots that talk about position and where the highest abundance binding sites are. And then we can generate a heat map. So we have protein area on the y-axis, we have DNA location on the x-axis, and then we're going to use a heat map to show the frequency. And so what we can do is we subdivide our molecules again. And so on this side, on the left side of your screen, you have the monomers, again, based on the parameters that we set forth with the conalbumin. And what we can see is on this axis right here, we just have the position. So the nucleotide position that I've shown on most of the y-axis that I've shown you before, or x-axis, I'm sorry. On this side, we have the area, and then the color dictates the frequency. So the more yellow or orange the color is, that's where the more frequent binding is seen. So if we then look for yellows and oranges, we see that in the monomeric size discretion, that the group that we made, there's a lot of binding here because it has the yellow or orange. And then there's also this secondary site right here. And if we cross correlate that with the sequence, what's interesting is that the published predicted site binds the monomers with some frequency, whereas the rest of the monomers bind to this novel site that I talked about when we talked about binding location. So if we then look at just the dimers that were more in line with what we would see for alcohol dehydrogenase, we can look at these dimers and we're looking at where the yellow and oranges are. And we can clearly see that the dimers bind with the highest frequency at this novel site, which we think is a pretty cool finding that has never been previously appreciated. So we're really excited to take what we've learned from using EM as one of the tools to really characterize this origin binding protein. So we've been successful at not only observing OR50 binding to the predicted sites, but we also were able to identify novel binding sites. And we're also able to differentiate where monomers and dimers might bind. And so we think overall that this will give us a lot of clues into understanding the function of this protein when we start adding other proteins into the mix. So if we have an additional protein, whether it's the processivity factor or the polymerase, is the binding site or the whether it's a monomer or dimer going to be more prevalent. In addition, we think that overall this dimerization most likely will probably stabilize the DNA protein interactions. And then we think that by stabilizing it at a specific location, then it can more efficiently recruit the rest of the DNA machinery, DNA replication machinery. So I have a few additional data slides to go through in terms of introducing a second protein. So this processivity factor And so a processivity factor is a protein, we have them in our cells. It just helps DNA polymerase stay attached to the DNA in order to continue elongating and copying that DNA. So it's an essential protein for KSHV DNA replication. It enhances the processivity of the polymerase, which is known as ORF9. It has already been shown to bind to not only the DNA, the origin DNA, but it also interacts with polymerase and RTA, which is that origin binding protein that I was just talking about in this earlier slides. And it is known to contain a dimerization domain. So again, like I was talking about at the beginning, EM allows us to see all these different structures that form. And we can take a step further and we can look at what these structures mean in terms of dimensions. So we broke it down into these two major complexes. And what we can do is we measured a subset. So this is more preliminary data. The OR50 story is a little bit further along. And we can measure the diameter of those structures. And we can also measure that internal pore. So what you can see is that it looks like it's protein, and there might be this empty center, which is consistent with the functions of processivity factors, which typically function as a sliding clamp, in which case they're going to hold on to the DNA, make sure that that polymerase still stays attached so it can still do its job. So it's interesting for us because the measurements that we were able to complete using the EM images allows us to understand that, in fact, that pore size would in fact accommodate a duplex DNA, which was about 2 nanometers in diameter. So our observations, our preliminary data observations with OR59 are pretty consistent with what's observed and what's published for EBV, this idea that it does form these oligomeric structures. There's been some contention whether it's only a dimer or if in fact it is oligomeric. We think that the head to tail dimers that were proposed for the EBV homologous protein actually happened because of head to tail dimers, and that could allow for the assembly into the structures that we think we're seeing, which are tetrameric and heteromeric. So 4 protein monomers coming together or 6 protein monomers coming together to form those structures. So we think that all of this together would help to bolster the idea that the processivity factor functions as a sliding clamp. And for this study, we are really excited to continue the experiments where we actually look at the protein and DNA together to see if these complexes are more frequent or if one of the two complexes, whether it's the tetramer or the hexamer, might be more stabilized because of the interaction with the DNA. So for future directions for our herpes DNA virus pre-initiation complex, we think that based on some of the data that I've shared with you today and some of the stuff that I haven't included, that RTA binds the origin as a dimer, and this binding as a dimer might in fact distort the DNA enough to allow some unwinding, and then some other of the accessory proteins, the single stranded binding protein and et cetera, can come in, and then that will allow for the recruitment of the processivity factor and then the polymerase. However, in the future, we really want to keep going with each individual protein characterization. So I told you that there was 8 essential KSHV DNA replication proteins. I only told you the story about 2 of them. So there's quite a few more that we still have to characterize individually, and that's the protein alone, as well as the protein, how the characteristics of the protein in the presence of the relevant pieces of DNA. We also really look forward to these combinational studies where we can look at multiple KSHV DNA replication proteins together in the presence and absence of the DNA sequences. Also, we have the ability to integrate any additional host proteins that we think might be also relevant to this process. And then all of that to really develop this idea of this in vitro DNA replication system. And once we have that in place, we could really start to identify and test for novel DNA replication inhibitors that will then go on to prevent viral DNA replication and then additional viral spread. So with that, I just want to thank the people that have helped me accomplish most of the data that I presented today. This work, like I said, started at UNC in the labs of Drs. Blossom Damania and Jack Griffith. And then we've continued this work, and the members of my lab, especially Jayla Calhoun. She's a recent master's thesis graduate student. She did a lot of that interesting analysis, looking at all the different variables, and was really great at taking some of that raw data, just EM micrographs, and then translating it into meaningful quantitative data. And again, I'd also like to thank my funding source, and I would welcome feedback on the project because we are definitely working on trying to get this published as soon as possible. And I'd also welcome any questions.
Thank you so much, Lindsay. Are there any questions?
Hello, Dr. Constantini. I thought the heat map for the binding sites was really interesting. Have you guys done the same thing but with the mutant protein to see how that would differ?
Great question. Yes, so we are in the process of doing some analysis. So we also have a truncated version of the protein. So I didn't go too much into it, but OR50, like all the different domains, have been a little bit worked out based on some previous published work. And so we have a truncation mutant that we have some preliminary data with, and we're hoping to do the same analysis to see if we're able to zone in, or does it make it spread out more? Does it get rid of the specificity? And so, yes, we are really looking forward to doing those studies, but we haven't done extensively point mutations or anything like that. But we do have a truncation mutant that we have preliminary data with that we're excited. So far, we're seeing it does maintain the truncation mutant that we have still has the ability to dimerize. And so we've been doing some of that analysis where we compare it with the other molecular protein comparative proteins to kind of get an idea of what the structure is. So yeah.
Thank you.
I think Ryan had his hand up, and I can also, I don't know if I can see the chat.
Yeah, hi. Thank you so much. This was a really great session. I'm so glad I was able to listen in. I have a question. So, when are you planning? You talked about how you're planning on doing further research, but do you have like an estimate time, like how many years? And also, are you planning on making a book into this? Because it's very interesting. This is also can be some really discovering breakthrough information that can really help just overall when dealing with viruses. So, are you planning on also turning to a book too?
Oh, well, thank you very much for all those wonderful compliments. I really appreciate it. So my first priority is we are in the works of publishing the OR50 studies. So in addition to the data that I summarized today, we have a bunch of different other data that supports this whole idea of this monomer and dimer having different preferential binding locations. And so our first goal is to publish that. And then incrementally, hopefully, as long as I have people who want to do research in my lab, the idea would be incrementally that we would publish a paper for each individual protein that we characterize. And then once we have some information based on how some of the individual proteins work in vitro, because you have to figure out like what's the right buffering conditions, what's the right stoichiometry. Once we start getting a better handle on all of that, we'll start doing the combinational studies. So in terms of characterizing the essential proteins that we have in our lab right now, we have about 5 of them purified and ready to go. My hope would be in like 5 years that we would be able to get that definitely done. And like the longer term, 5+ years would definitely be working on getting that DNA replication system in vitro, get finalized and going.
Great. Thank you so much.
There is a question from the chat I can read for you.
Great, thank you.
Okay, so Zoe asks, when you started your research, were you expecting to find as much as you have? This research is incredibly interesting and should be shared on a grander scale. What impacts are you hoping this will have on society and the general public? Wow, that's a question.
Oh, okay. Well, I'll take it bit by bit. So if I flash back to the start of my postdoc, no, I had no idea that we would be able to get this wealth of information looking at just purified proteins and purified DNA, because I was much more used to looking at entire cells and fluorescent microscopy and labeling different proteins and looking at live cell protein dynamics. So I was like, I don't know about this because EM is basically a snapshot. Nothing is alive, you're fixing it all. I was like, I don't know how much information we can really get out of this, but being part of the Griffith lab really showed me how powerful it was. So he's a very senior PI and he's been doing EM like this for decades, and he's made some amazing discoveries. He did a lot of work visualizing the first histone chromatin complex, looking at how the histones bind around are bound by DNA, and looking at them with very similar methods to the ones that I was talking about today. So I was really excited, but I didn't really know how much I was going to learn. And I'm so pleased and so excited that I've been able to transfer this to NCCU. So one of the major hurdles is having a microscope. So not a lot of new faculty are going to come in with an electron microscope. And it just so happened that Dr. Griffith had multiple microscopes and he wasn't using one of them. And so he donated that to me when I moved to NCCU. So that makes this research possible. If I didn't have a microscope, I would be going back and forth to UNC all the time, and that would make the practicality of having research and graduate students work on this a little bit more hard and difficult. But I'm very fortunate that I can train students here to do the EM methods that I've shown. So no, at first I did not have all of this crazy expectations in my mind. I was just really excited to get into virology and learn more about EM, and it's just been a great project. I've been very fortunate in that sense that I've been able to take a microscope from a different institution and have it here and be able to push this project forward. I also like it for the context of a lot of undergraduates and graduate students have a frame of reference about DNA replication in cells in general. So now I'm just translating very similar proteins, but the virus specific ones. So I like that interface of knowing what happens from your cell biology and molecular biology classes and then applying it to a virology question. And so I do think it's a long ways away, but if we can get to the point where we have that in vitro system and we can then do some screens to see if we can disrupt it, I do think that we could learn ways to reduce KSHB viral spread. Also, these proteins are really homologous to EBV, which is a much more prominent virus in the human population. So we could apply these same insights that we learned from KSHB to EBV as well, which I think will also have a broader impact. Yeah. Now I think I can see the chat now. Kaylee had mentioned, oh, not a question. That she was very excited about learning more about viruses. Okay. Well, I'm glad that you enjoyed the talk, and I hope that I hope that it was in a way that most people could follow. And Gloria, is there a difference on how well the dimers versus monomers bind onto different sites? Is there more efficient binding at one site than the other? Like the dimer is found more bound. So that makes me think that it's more efficient in binding. Yes. So we're trying to do a little bit of those studies. So the question is really getting at, is the binding of the dimer stronger, and that's why we're seeing higher frequency. We are thinking about experiments to actually test that. We do have a setup where we use fixed and non-fixed reactions, and the idea being that in the non-fixed situation, you are only really preserving those visualizing those interactions that might not be as prevalent. Whereas if you fix it, you just are fixing everything, whether it's predominant or a more efficient binding. So we are doing those comparisons, and we might need to do like more of like ELISAs or some sort of kinetic based things to kind of think about that as well.
I think I might have another question. I might have misunderstood what you said, but you said you found a new binding site and it was an ORF, right? Site for ORF protein or something like that?
So for the ORF50, the origin binding protein, we identified a novel site, yes.
Yes, so usually that ORF is because it's an open reading frame. Have you thought about like what is associated there, what the transcript might be, like why is that happening? Because from the data you were showing, I think that had more binding than any other site that was already identified.
Yeah, exactly.
So
and
what
so
is
right.
that?
The novel site had more dimers and the novel site had the most protein bound. And so what that means is we are actually looking at the DNA sequences and seeing if, because the origin of DNA replication, not only important for DNA replication, so that's where the bubble is gonna open and allow DNA polymerase to come in and copy, but it also corresponds with some promoter sites for some pretty highly regulated or highly expressed lytic proteins. So we're trying to do some sequence analysis to see if there's, based on stuff that's published and doing a comparison to see if this novel site might have a more of a transcriptional activity. and kind of looking at that literature separately and seeing if there's more of a consensus sequence that has already been identified as a transcriptional role, and maybe within that novel site, we're going to see that activity as opposed to the DNA replication activity.
Thank you.
Yeah. Well, if anybody has questions after the fact and they want to email me, Rachel has my email, so I'm happy to answer them that way as well.
Great. Thank you so much. We had some great questions, and
Yeah, those were awesome.
we appreciate that you took the time to talk to us today.
Well, thank you for this opportunity. It was nice to see everyone. I hope you have a good rest of your semester.
Thanks. You too.
Thanks, Lindsay.